So, hi students, in this class we will be discussing about regulation of cholesterol biosynthesis. So, if you have not seen the cholesterol biosynthesis steps, you can pause this video, you can go back and watch that and then come back here. You can find the link in the description. Now, the thing is, regulation of cholesterol biosynthesis is more important than the steps of cholesterol biosynthesis. Number one, because these days a detailed stepwise question doesn't come. Mainly the question that will come in theory is regulation up to mevalonate, alright. But in case of Viva, you can expect any question, any step if you are answering that good. So we'll be going into the regulation of cholesterol biosynthesis, which is actually a topic for number one, a long question, number two, Viva, number three, short note, and anything you can imagine. Okay, so if examiner wants to ask you about cholesterol synthesis, there will be the questions will be in line of what is the precursor of cholesterol biosynthesis. The moment you tell acetyl CoA, he or she may go into TCA cycle, or he or she may go into the cholesterol synthesis proper, and that question will immediately follow with regulation of cholesterol biosynthesis, because the examiner will stop you. If you start saying all the steps, acetyl CoA plus acetyl CoA becomes acetoacetyl, then it becomes HMG CoA, then it from HMG CoA reductase acts on it, then examiner will stop you. Fine. Now tell me how cholesterol biosynthesis is regulated. Okay. So basically, as I told you, and you already know this because this is a revision topic, HMG CoA reductase is the main key enzyme which needs to be regulated in order to either stimulate or inhibit the body need for cholesterol biosynthesis. Because when we are discussing HMG CoA reductase, we are discussing about the endogenous cholesterol biosynthesis, not the cholesterol that we receive or eat from outside food or junk food. Alright, so this is the key regulator enzyme. So the few things about this HMG CoA reductase enzyme, its half-life is 3 hours. Okay. So if any short-acting inhibitor will be able to inhibit this cholesterol synthesis key enzyme, that is HMG CoA reductase, there are many enzymes which the half-life of which are mainly very high. Alright. So I don't know how many of you are acquainted with these drugs. So lovastatin, simvastatin, atorvastatin. Generally, they are given in once daily dose. Okay, leave it apart. Even this information is not a must know area. Okay, half-life is 3 hours. You can jolly well forget. However, what is important is this degradation. Okay, it depends on the cholesterol levels. So if the cholesterol is very high, okay, this enzyme degrades very fast. Why? Simply because high body cholesterol does not need production of any more cholesterol. You get my point? So this is kind of a feedback inhibition. So if the blood cholesterol or the body cholesterol levels are low, then this enzyme, we need more and more cholesterol, so we need more and more HMG CoA reductase, so this will not be degraded that fast, so enzyme will remain in the system. Okay, this is a concept, but the main thing, the main key points in your answer, the main headings will be this one. Number one, hormonal. Okay, the thing I just discussed is feedback. Number two, covalent, and number three, feedback inhibition. So this is not only true for HMG CoA reductase, but this is true for any enzyme. So be it phosphofructokinase or be it regulation of fatty acid synthesis, beta oxidation, try to frame your enzyme regulation or any step regulation under these three headings: hormonal, covalent modification, mainly phosphorylation and dephosphorylation, and lastly feedback inhibition. In some cases there may be an allosteric component, but allosteric component is not there in cholesterol biosynthesis. So the thing is, I already told you in brief in the last class the role of insulin and glucagon phosphorylation and dephosphorylation. So we'll be elaborating those concepts only, and the class will be over very soon. I hope so. Anyway, so see, there are things you should remember. Insulin in well-fed state stimulates and increases HMG CoA reductase. I already told you insulin induces synthesis of cholesterol. It increases cholesterol biosynthesis. So insulin will stimulate cholesterol synthesis by increasing or stimulating the HMG CoA reductase enzyme. Anyway, I discussed in last class the mechanism of phosphorylation and dephosphorylation. I told you anabolic process, insulin, etc. Can you tell me, if insulin is stimulating HMG CoA reductase, this enzyme, in which form this enzyme will be active, phosphorylated or dephosphorylated? This is an anabolic enzyme, mind it.
Dephosphorylated.
Absolutely right, absolutely right. Okay. So let's see, let's go forward, but the answer is right. Okay, this thing I will be discussing again, but you are absolutely right. So the basic concept is same for all: carbohydrate, protein and lipid metabolism, but mind it, lipid and carbohydrate metabolism is not in the 25th ka viva. Okay, but trust me, you shouldn't forget them. Okay, till 25th don't study them. Definitely you should save your time in studying anatomy. I cannot comment about physiology yet. Uske baad you should revise everything. You start revising on your own. Just make a routine. I'll also tell you how to revise optimal time. Kaise you can utilize in revising everything. Those classes will happen. Anyway, so see. So if insulin does one thing, of course glucagon and glucocorticoid will do the reverse. So naturally this is a no-brainer that it will inhibit HMG CoA reductase and it will decrease cholesterol biosynthesis. It means that the phosphorylated form is inactive. Okay, so insulin stimulates, glucagon inhibits. So if you are out of time, okay, these are the keywords. Believe me, a student who is out of time may score more than 60% in a long answer, even if the long answer is in 4 to 5 lines. Nowadays, not nowadays, every time, other than history exam, examiner always searches for keywords and key concepts. Okay, so if you mention the keywords in 1 or 2 lines, and more important, if you draw a flow diagram, you don't need to write anything in descriptive. Okay, so just mention this one: insulin stimulates and glucagon inhibits. So this is number 1. You can give any diagram. Okay, this is the diagram of Leninger. Okay, there is too much going on here. Just note this step. This is the step of HMG CoA reductase. Okay, hydroxymethyl glutaryl CoA, and this is the HMG CoA reductase enzyme. And insulin is stimulating and glucagon is inhibiting. Okay, there are many more steps which have been shown in this diagram, but for you, I am only elaborating this part now. Insulin is stimulating and glucagon is inhibiting. So just write this reaction: HMG CoA. The enzyme is HMG CoA reductase and the product is mevalonate. Insulin give a plus sign and glucagon give a minus sign, and you are done with hormonal regulation, right? Okay. So now we go to the covalent modification of regulatory enzyme HMG CoA reductase. So the main covalent modification that you need to know is phosphorylation and dephosphorylation, and I hope you already have an idea what is going on here. So short-term regulation of cholesterol biosynthesis is by phosphorylation and dephosphorylation of key enzyme HMG CoA reductase. You may jolly well forget, bhul jao short-term or long-term. Just remember, this is actually for PG. Okay, if you are a PG student, then ये जानना है. Covalent modification से short-term regulate होता है. In the long-term, covalent modification gets reversed. छोड़ दो. So this is the key enzyme HMG CoA reductase. So क्या होगा? You already told, phosphorylated form is the inactive form. Absolutely right. And dephosphorylated form is the active form. This is actually related closely with insulin and glucagon, but in general we separate the two things, more or less the same thing. But in hormonal, you don't explain the detail that hormone insulin will be phosphorylating. No, you leave that part under a separate heading of covalent modification. Okay, so just like that, वहाँ पे था insulin stimulates, glucagon inhibits. Over here, dephosphorylated active and phosphorylated inactive form. Now you need to give a bit of diagram. Okay, and this actually is a two-step regulation. So abhi you can take a screenshot, you can see your book, or you can draw on your own अभी ये देख के. हाँ देखो, see the HMG को reductase, the OH form means dephosphorylated form, okay, which is active, and this P is phosphorylated form. Now tell me, phosphorylation is done by what class of enzyme? Glucose to glucose 6 phosphate. कौन सा enzyme करता है? Who will be doing this? Hexokinase or glucokinase. All right, so absolutely right. So kinase group of enzyme, that is a transferase, catalyzes all phosphorylation reaction. So यहाँ पे भी there will be one kinase. Which what kinase? This one, AMP activated protein kinase. This is the enzyme that phosphorylates HMG CoA reductase and converts the active form to inactive form, right? In turn, okay, now this AMP dependent protein kinase also needs to be activated. This is the same mechanism which you have studied for other enzymes in carbohydrate metabolism. Okay, the kinase also needs to be activated, and this activated kinase is actually, this is confusing. Okay, please listen. HMG CoA reductase is active in dephosphorylated form. Okay, this is, I told the right thing but I wrote in wrong area. This is active in dephosphorylated form and it is inactive in phosphorylated form, right? However, this AMP dependent protein kinase, okay, this one is active in phosphorylated form. Remember, we are talking about the protein kinase enzyme that has also an active and inactive form. So this, since this is active in phosphorylated form, naturally there will be another kinase who converts this dephosphorylated form to a phosphorylated form. Always remember, something, a substrate to a substrate phosphate is done by kinase, and a substrate phosphate to substrate is done by phosphatase. You already know that, okay, because neoglucogenesis, the enzyme was glucose 6 phosphatase, which is the direct reversal of hexokinase enzyme. So over here same, this HMG CoA reductase, this phosphorylated form will be converted back to the active form under the help of phosphatase. And who will be stimulating this phosphatase? Insulin. Same thing happens over here. Dekho, insulin stimulates phosphatase, okay, which actually deactivates this protein kinase, okay. So what happens? This deactivated protein kinase, okay, low activity, cannot convert this active form into an inactive form. Just tell me, समझ में आया? क्या समझ में आया? Right. So basically it's an indirect step. हम लोग क्या इतने देर से what we were of the impression that glucagon is converting dephosphorylated to phosphorylated, वैसे नहीं होता है, होता है. The thing is, देखो, so indirectly अब क्या होगा? When glucagon activity is high, this will activate this AMP dependent protein kinase, which in turn will inactivate HMG CoA reductase. एकदम सही बोले. हम लोग इंसुलिन को देख लेते हैं. इंसुलिन कैसे करेगा? इंसुलिन हैज टू एक्टिवेट दिस वन, लेकिन बेसिकली इट इज इनहिबिटिंग द कन्वर्शन ऑफ एक्टिव टू इनएक्टिव. कैसे? इंसुलिन इज इनहिबिटिंग द प्रोडक्शन ऑफ एक्टिवेटेड प्रोटीन काइनेज. सो ये इसका एक्टिविटी अगर कम हो जाएगा तो इसका एक्टिव से इनएक्टिव कन्वर्शन भी कम हो जाएगा. सो बेसिकली इट्स अ 2 लेवल कंट्रोल. आई एम नाउ इरेजिंग एवरीथिंग एंड एकदम क्लियर पिक्चर दिमाग में रखना. This is very important, and remember, as I told you, writing this answer is very easy. Just a diagram समझ के या नहीं समझ के theory copy में उतार दिया तो examiner is bound to give you full marks. लेकिन बोल के समझाना is very tough. You need to have absolutely clear concept that HMG CoA reductase active form is dephosphorylated. लेकिन वो फॉस्फोराइलेट हो जाएगा अगर प्रोटीन काइनेज एक्टिविटी हाई रहा तो. प्रोटीन काइनेज एक्टिविटी कब हाई रहेगा? ग्लूकागॉन अगर हाई रहा तो. तो इसमें इंसुलिन क्या करेगा, इंसुलिन ये एक्टिवेटेड प्रोटीन काइनेज को ही नहीं बनने देगा। अगर ये नहीं बनेगा तो जो एक्टिव है फॉर्म डीफॉस्फोराइलेटेड वो एक्टिव ही रहेगा। ओके, सो दिस इज द हार्मोनल रेगुलेशन, हार्मोनल नहीं, कोवैलेंट मॉडिफिकेशन रेगुलेशन ऑफ cholesterol biosynthesis. It is tough, but now that you have studied through carbohydrate metabolism and protein metabolism, and this one you already know. So देखो, the same thing I am repeating over and over again. Under the influence of hormone insulin, what will happen? The HMG CoA reductase is dephosphorylated, right? Which activates HMG CoA reductase and increases cholesterol biosynthesis. So there is no problem in understanding this slide. ठीक है? So the exact reverse thing will happen in case of glucagon. Glucagon is phosphorylated by CAMP dependent protein kinases. This will lead to inactivation of HMG CoA reductase and ultimately this will inhibit cholesterol biosynthesis. So ये दो slide में we have combined both the hormonal and the covalent modification. Okay. Now apart from that, you already know that insulin अपनी जगह पे है अकेले, and there are other hormones that also acts like glucagon, right? So see, glucagon, sterols, glucocorticoid, and ये वाला बहुत important है MCQ के लिए, low ATP level, it inactivates HMG को reductase, मतलब energy बहुत अगर कम है, okay, ATP level is, if it's very low, then cholesterol synthesis will also be hampered. Why? Because this is an active process. The main thing is it's an active process. पिछले हफ्ते का अगर process देखते हो तो you will see there are multiple processes in which residue is phosphorylated. Pyrophosphate बनता है एक, ठीक है ना? So that needs an ATP molecule. However, this is important. Thyroid hormone. In spite ये catabolic hormone, normally it is a calorigenic hormone, लेकिन यहाँ पे it acts like insulin. High ATP level you already know by now. वहाँ पे low ATP level was inhibiting, therefore high ATP level will activate. This is the key thing to remember. Okay, thyroid hormone actually activates, मतलब increased thyroid hormone will activate HMG CoA reductase. So if you are asked the factors that affect HMG CoA reductase, definitely insulin, glucagon and other hormones and ATP level. Other hormones में क्या याद रखना है, जो catabolic hormones है, glucagon, glucocorticoids, growth hormones, steroids, they all inactivate HMG CoA reductase, and insulin and thyroid hormone in this case they activate HMG CoA reductase. तो ये तो हुआ hormonal and covalent, ठीक है ना? अब देखो, the third was feedback inhibition, right? So what is feedback inhibition? As simple as that. ज़्यादा है body में तो और नहीं चाहिए. So sufficient amount of body cholesterol can regulate its own biosynthesis by inhibiting HMG CoA reductase. Very easy. बॉडी में बहुत कोलेस्ट्रॉल है, बोलेगा कि भाई अभी कोलेस्ट्रॉल नहीं चाहिए, so please अपना काम बंद करो. Very simple. However, ये भी you should know that ingestion of cholesterol, बाहर से अगर कोलेस्ट्रॉल हम लोग ले रहे हैं तब भी बॉडी कैन सेंस, and that will also inhibit endogenous cholesterol synthesis. You may feel when you eat cholesterol from outside, it will inhibit the cholesterol synthesis from inside. Okay, and I already told you that endogenous cholesterol synthesis predisposes the body to heart diseases. Okay, however, you should know that when you are eating a lot of cholesterol from outside, you are already contributing to the body cholesterol. Okay, so it is not wise to inhibit the cholesterol synthesis in this way. However, you should know that when you are eating too much fat-rich food or cholesterol-rich food, body will sense that we are getting excess cholesterol from outside, and that also will inhibit cholesterol synthesis. Not only it will control at the hormonal level, I mean protein synthesis level, it will be controlled at transcription level. What is transcription level? You are already undergoing transcription classes, but the gist is whatever DNA we are having in our genome and whatever enzyme we are needing, we actually transcribe those mRNA, and those mRNA transcribe those protein. In this case, the proteins are the cholesterol synthesis enzyme of cholesterol synthesis. So if we inhibit the synthesis of mRNA, then this cholesterol synthesis enzymes will be inhibited. Remember, we are not giving any enzyme inhibitor from outside, or we are not doing any feedback inhibition. We are actually influencing at the genetic level. mRNA synthesis ही बंद हो गया, then there will be no translation. No translation means there will be no protein. The proteins in this case are the enzymes, and this thing is actually controlled by this one, sterol regulatory element binding protein. This name you should at least know. This name you should mention in your oral answer, and this name you should mention in your theory script answer also. So rate of HMG CoA reductase mRNA synthesis is controlled by transcription factor SREBP. So SREBP you should write, and you should underline in your answer. If you are writing transcription control, just mentioning this SREBP is enough, right? But there are few exceptionally good students who want their answer scripts thoda hatke and above average. Especially for those of you, you can give a diagram which features SREBP. Okay, now this diagram may not be present in your textbook, right? So you can draw this diagram in a simplified way, or you can draw this exact figure. Trust me, you don't need SCAP and S1P. You don't need them. Just you should draw a gene, okay? And SREBP is the transcription element, and everything is controlled at the nucleus. So the thing is, all the foods and sterols, they inhibit this SREBP. If you are not understanding this diagram, this is not your fault, because basically you need a bit of transcription level training in genetics. So what to do? भूल जाओ इसको. I can make you understand, but after 25th का भाईवा, ठीक है? I will be discussing SREBP in detail. अब के लिए इसको दिमाग में मत घुसाओ. Even if you are a exceptionally good student, just forget this, because इसको समझने में जो टाइम जाएगा उतने टाइम में you can go through your anatomy lessons. Okay? So we come to drugs or medicines. This is the main reason why we are starting, or we are learning cholesterol biosynthesis and its regulation in the first place. So to make the class interesting, I could have started with these, okay, pharmacology class, and trust me, it would have happened in that way. We could have integrated the class with pharmacology and medicine, jahan pe they would have come and showed you a patient ka photo and report, and then they would have shown उसका prescription. उसका prescription में तुमको ये सारा ड्रग तो दिखेगा ही. Any patient, okay, of heart disease or high cholesterol must have this statins. Okay. अब देखो, these are competitive inhibitors. Competitive inhibitors का feature क्या है? Feature is substrate concentration अगर बढ़ जाए तो कॉम्पिटिटिव इनहिबिटर काम अच्छे से नहीं करेगा. इनहिबिशन कैन बी ओवररूल्ड, ओके, एंड इनहिबिटर कॉन्सेंट्रेशन बढ़ाते रहो तो सबसेट कॉन्सेंट्रेशन एंड इनहिबिशन ज्यादा से ज्यादा बढ़ते रहेगा. दिस इज बेसिकली कॉम्पिटिटिव इनहिबिटर बिकॉज दे कॉम्पीट फॉर द सेम एक्टिव साइट, है ना. सो जस्ट नो दिस दैट ड्रग्स लाइक लोवास्टैटिन एंड सिम्वास्टैटिन आर कॉम्पिटिटिव इनहिबिटर्स ऑफ HMG CoA reductase, and they decreases the endogenous cholesterol biosynthesis. कैसे करेगा? Basically lovastatin is converted in vivo to this one, okay. Pharmacology question, but since we are integrating ये देखो, mevaloninic acid is actually the active principle, but trust me, if you forget this slide and ये वाला only बोलते हो, lovastatin, simvastatin and atorvastatin. इसका brand name you need to know, Storvas या Atorva. How many of you have come across patient जिसका prescription में ये सारा दिया हुआ है, Storvas या Atorva? मालूम है?
नो सर.
आज से जान लो तो मालूम पड़ गया. हर चीज सबको मालूम नहीं होता, तो आज से सीख गए, ठीक है ना. कहीं भी हाई कोलेस्ट्रॉल या ओबेस पेशेंट चेंबर में आए, उसको एक स्टैटिन चिपकाना ही है प्रेस्क्रिप्शन में. इट इज अ मस्ट. इट विल सेव यू इन द कोर्ट. ठीक है ना. अगर उसका ओबेस कंडीशन है एंड यू डोंट प्रेस्क्राइब स्टैटिन एंड लो डोज एस्पिरिन, यू मे बी ट्राइड इन अ क्रिमिनल कोर्ट. सो ये मिस नहीं करना है. So the thing is, the HMG reductase enzyme and the structure of atorvastatin, they actually mimic each other. ठीक है ना. I mean HMG and atorvastatin, they mimic each other क्योंकि the enzyme is HMG reductase, the product is mevalonate, so दोनों का structure same है, so they competitively compete with each other. And इसीलिए you will also find that there are multiple statins that will be given to a patient, okay, because if the patient will also be asked to modify his or her diet. मतलब क्या है, हम लोग स्टैटिन भी खा रहे हैं आराम से सारा दिन पिज्जा बर्गर खा रहे हैं तो उससे होगा नहीं कुछ काम. व्हाई? बिकॉज कोलेस्ट्रॉल बायोसिंथेसिस HMG रिडक्टेज इज मोर एंड मोर, then if HMG is more, then the amount of statin dose will fail. So you need to ask the patient to modify his diet, exercise and उसके साथ statin देना होगा, mainly because this is a competitive inhibitor. अब देखो statin कैसे काम करेगा. These are the effects of statins. The cells, this thing, short note on atorvastatin is very common. ठीक है ना. तो ये सारा लिखना ही पड़ेगा. All these things you need to know. ये अगर नहीं भी लिखे तो भी चलेगा. Number 1, you should write competitively inhibits HMG reductase, okay, a key enzyme for de novo biosynthesis of cholesterol. And what are the effects? Atorvastatin से क्या होता है? What happens? The cell expresses more LDL receptors, decrease in serum LDL level, increased HDL level, improved or HDL to LDL ratio बढ़ जाता है, which is good for the body, and suppresses production of VLDL in liver. Basically what it does, it reduces bad cholesterol and it increases good cholesterol. HDL is good cholesterol, you know na, HDL is good cholesterol. Why HDL is good cholesterol? Tell me.
More protein.
What? Why we say LDL वैल्यू ज्यादा होने से प्रॉब्लम है, दिक्कत क्यों है?
Because वो फॉस्फोलिपिड और वो लिवर से एक्स्ट्रा हेपेटिक टिश्यू में ले जाता है.
Absolutely right. So the problem is liver में कोलेस्ट्रॉल है, it's fine, but when it will take to extrahepatic और peripheral tissue, मतलब arteries, तभी जाके हार्ट अटैक, स्ट्रोक ये सब होगा. So LDL level अगर ज्यादा है तो extrahepatic tissue में cholesterol deposition will be more, and HDL does the reverse thing, it does reverse cholesterol transport. ये peripheral tissue से लेके liver पे पहुंचा देगा. liver पे we have no problem in accumulating cholesterol, therefore HDL is a good cholesterol because it prevents all cardiovascular accidents. Now this thing you should know for your future in chamber, and I am not discussing in detail. देखो, this is actually very well tolerated drug. जो भी पेशेंट को दोगे कुछ प्रॉब्लम होगा नहीं. only problem is if the patient is already having hepatotoxicity. मतलब पेशेंट को हेपेटाइटिस बी, हेपेटाइटिस सी, अगर अमिताभ बच्चन इज हैविंग हेपेटाइटिस सी, मालूम है क्योंकि उनको कूली मूवी में उनका स्प्लेनिक रप्चर हुआ था, फिर उनको 29 यूनिट्स ऑफ ब्लड ट्रांसफ्यूजन दिया गया. तब के जमाने में देयर वाज नो स्क्रीनिंग बिफोर ब्लड. आज अभी ब्लड विल बी टेस्टेड फॉर हेपेटाइटिस बी, हेपेटाइटिस सी एंड HIV, HBV, HCV एंड HIV के लिए ब्लड इज ऑलवेज टेस्टेड बिफोर ट्रांसफ्यूजन. तब के जमाने में नहीं था एंड उनको वही ब्लड ट्रांसफ्यूजन से हेपेटाइटिस सी हो गया था एंड ही इज स्टिल सफरिंग फ्रॉम हेपेटाइटिस सी. उनका लीवर 80% काम नहीं करता है एंड ही टेक्स अराउंड 20 मेडिसिन पर डे. उनको एक और भी बीमारी है, दैट इज माइस्थेनिया ग्रेविस. अमित जी इज अ वेरी सिक पर्सन बट इट डजंट अपीयर ऑन स्क्रीन. एनीवे, so remember, if he or she is having hepatotoxicity, मतलब patient is an alcoholic, cirrhosis है, तो you cannot give statin or you need to give other statins which are mobilized by kidney, not liver. There are other statins which will be taught in pharmacology. दिक्कत जो है वो है मायोपैथी, मसल पेन होता है. स्टैटिन का अगर बहुत हाई डोज होता है ना तो पेशेंट विल ऑलवेज कंप्लेन ऑफ मायल्जिया, सब ऑलवेज लगेगा कि बॉडी एक हो रहा है, बदन दर्द. सो दैट यू नीड टू कीप इन माइंड. एंड द थिंग इज इट इज वेरी एक्सपेंसिव. ठीक है ना. अगर ओबेस प्रेग्नेंट पेशेंट आए इवन विथ सिम्टम्स ऑफ कोलेस्टेरॉल, उनको कभी भी स्टैटिन नहीं देना है. ठीक है ना, इट इज कॉन्ट्राइंडिकेटेड अगर पेशेंट इज अंडरगोइंग मतलब डूइंग लैक्टेशन, मतलब पेशेंट इज फीडिंग द बेबी, ठीक है ना, उनको भी नहीं देना है बिकॉज इट इज कॉन्ट्राइंडिकेटेड इन बेबीज एंड फीटस, वेरी इंपॉर्टेंट, ये जानना है एज अ जनरल नॉलेज, ठीक है ना. सो हैव यू गॉट एनी आइडिया व्हाट इज xanthoma और xanthelasma, मालूम है? बोलो तो. ओके देखो, एक ही लाइन और बचा है. बाइल सॉल्ट इनहिबिट्स इंटरनल इंटेस्टिनल एचएमजी को रिडक्टेज, ये एक लाइन लिख के रख दो नोट्स में. बाइल सॉल्ट विल इनहिबिट इंटेस्टिनल एचएमजी को रिडक्टेज. Okay, this is one line you should add at the end of your answer. Okay, so we stop our discussion till here and we'll continue with cholesterol transport in our next class. Till then, bye and take care.